Okay, good day everyone. My name is Professor Carl J. Brigante. I'm a full-time faculty member of the College of Medical Technology. I am also your microbiology lab coordinator for this subject, microbiology. I'll be discussing two lessons. The first lesson is all about cell structure, and the second lesson is all about taxonomy and microbiologic techniques, the basic microbiologic techniques and its principle. To begin with, let's discuss cell structure first. For this PowerPoint presentation, let's discuss cell structure. So, I know you have already idea of the cell structure because this is being discussed to you on your biochemistry and physiology. So again, according to the scientists, that the world is split into two kinds of organism. You have your prokaryotic organism and your eukaryotic organism, which have two different types of cell. An organism can be made up of either one type or the other. Some organisms consist of only one measly cell, but even so, that the cell will either be prokaryotic or eukaryotic. The difference between eukaryotic and prokaryotic cells has to do with little stuff doing of the cell called the organelles. So your prokaryotic cells are simpler and lack eukaryotes membrane-bound organelles and nucleus, which encapsulates the DNA structures. Okay? Although more primitive than eukaryotic, prokaryotic bacteria are the most diverse and abundant organisms on Earth. We humans are literally covered in prokaryotes inside and outside. So we have normal flora. On the other hand, all humans, animals, plants, fungi, and protists are eukaryotic organisms. And though some eukaryotes are single-celled, like your protist organism, there are no prokaryotes have more than one cell. So, let's discuss the two separately. So, we'll start with eukaryotic organisms. So, the plant cell and your animal cells, as well as the protist organisms. Slide, you'll see there endoplasmic reticulum. So, endoplasmic reticulum is a highly convoluted system of membranes that are interconnected and arranged to form transport network of tubules and flattened sacs within the cytoplasm. So you have two types of endoplasmic reticulum. So you have there the rough endoplasmic reticulum, which contains your ATS subunit ribosomes. Remember, since this is eukaryotic organisms, it contains ATS, but per prokaryotic organism, it is 70S. Okay, so your rough endoplasmic reticulum contains major producer of your glycoproteins and produces new membrane materials that is transported throughout the cell. Your second type of endoplasmic reticulum is your smooth endoplasmic reticulum. This is smooth because these do not contain ribosomes. So, the main function is to synthesize lipids and some aspects of carbohydrate metabolism. So, you have there microscopic pictures of your rough endoplasmic reticulum, rough because of the ribosomes presence, and your smooth endoplasmic reticulum, absence of ribosomes. Another cytoplasmic structure is your Golgi complex apparatus or body. So, this organelle is the only organelle named after the discoverer. So, I want you to find who is this discoverer. That's additional note. Okay? So, your Golgi complex communicates with ER. Your Golgi complex consists of stacked membranes that function in concert with your endoplasmic reticulum to chemically modify and sort products of the endoplasmic reticulum into those destined to be secreted and those that functions in other membranous structures of the cell. So, Your Golgi apparatus or Golgi bodies is a factory that receives proteins from the ER and then further process. Pag receive niya, ini-sort out for transport, and then eventually, those proteins will be sent to lysosomes, to plasma membranes, and for secretion. So, basically, your ribosomes are called as packaging plants of the cell because this helps in process and package proteins and lipid molecules destined to be exported from the cell. So as I said, it will be exported to lysosomes, to plasma membranes, or secretions. So another cell structure is your mitochondria. So mitochondria, singular, mitochondrion, these are known to be your powerhouse of the cell. Okay? So these are double membrane bound organelles with a typical size of 0.75 to 3 micrometers. So these are usually found in most mammalian cells. Exceptions are your mature erythrocytes. Remember, your mature erythrocytes do not contain mitochondria. Classically, your mitochondria, as I said, are called your powerhouse of the cell because these are the major site of ATP synthesis and are therefore exceptionally important to function both microscopically and macroscopically. However, class, there are recent studies that there are some major functions of your mitochondria. So I posted here the five basic functions or roles of your mitochondria. Okay? So, numbers of studies had been demonstrated that your mitochondria can function according to production of ATP, store of calcium, regulation of innate immunity, apoptosis, and stem cell regulation. So, if you want to read that one, there's a link posted below. So, I want you to read that one. But let's just discuss a little bit. So, your production of ATP. For production of ATP, perhaps you already know that your mitochondria is major function is to produce ATP, which is the energy currency of the cell. This complex multi-step process is essential for proper functioning of the body, and this function can contribute to a variety of disease, like ranging from diabetes to Parkinson's disease or to more rare genetic disorders. So I know you already know that mitochondria helps in production of ATP. For calcium, so your mitochondria class helps in calcium storage, particularly for calcium hemostasis. So mitochondrial calcium exchange is the flow of calcium in and out of the cell of a mitochondria and process in metabolic regulation and cell death. So there are recent studies class identify that the calcium efflux from mitochondria plays a vital role in heart functions. So it knocks down mitochondrial sodium-calcium exchanger, which associated from sudden death due to heart failure. Your mitochondria also helps in regulation of innate immunity. So innate immunity is the born system that recognizes, the response to infections by pathogens, providing immediate, non-specific defenses. So mitochondrial proteins, which is known as mitochondrial antiviral signaling proteins, plays a key role in innate response, which is important during viral infections that helps induce antiviral and anti-inflammatory pathways. Your mitochondria also plays a role in programmed cell death or your apoptosis because it stirs caspases. So your caspases, by the way, are family of protease of enzymes that plays important role in programmed cell death. So you will know more about this enzyme in your pathology subject. So Apoptosis, again, is a highly controlled process of programmed cell death which is used by multicellular organisms. So, mitochondria control the intrinsic and extrinsic release of proteins such as cytochromes from their entire membrane space in response to cell stress, like heat, infection, hypoxia, increase of calcium, and nutrient deprivation. So, disturbances to these stresses regulates or associated with the development of disease like cancer, tissue damage, and some of them follow stroke. And lastly, your mitochondria helps in stem cell regulation. So, there are studies that mitochondria are thought to play crucial roles in maintenance of pluripotencies, differentiations, and reprogramming of induced pluripotent stem cell. So, the generation of your reactive oxygen species by mitochondria has been shown to regulate somatic stem cell fate. So, more of the readings class, I want you to read that one on this link. So your mitochondria has three main structure. You have your outer membrane, your matrix, and your inner membrane. Your outer membrane contains proteins known as porins, which allows movement of ions into and out of the mitochondrion. So the enzymes involved in the elongation of fatty acids in the oxidation of adrenaline can also be found in the outer membrane. Again, enzymes involved in the elongation of fatty acids and oxidation of adrenaline can also be found in this part of your mitochondria. Matrix, your matrix class is the space within your inner membrane of the mitochondrion that contains enzymes of the Krebs cycle and fatty acid cycles. So, alongside with your DNA, RNA, ribosomes, and calcium granules. So, this membrane class, your mitochondrial matrix or your intermembranous membrane, surrounds the mitochondrial matrix where the citric acid cycle produces the electrons that travels from one protein complex to the next in the inner membrane. At the end of this electron transport chain, the final electron acceptor is oxygen, and this ultimately forms water. At the same time, the electron transport chain produces ATP. That is why they are called oxidative phosphorylation. So I want you to review your biochem. That's why I posted here. Inner membrane, the third part or the third structure, contains variety of enzymes, particularly your ATP synthetase, or ATP synthase rather, which generates ATP in the matrix and transport proteins that regulates the movement of metabolites into and out of the matrix. The inner membrane is arranged into crestae, which is an inner folded membrane, which the main function is to increase the surface area availability for energy production via oxidative phosphorylation. So what is oxidative phosphorylation? Remember, in the mitochondrion class, what proton gradient does is to facilitate the production of ATP from your ATP. Oxidative phosphorylation is when your ADP is phosphorylized to ATP, which is dependent on oxidative reactions, which is very dependent on oxidative reactions. So, how many mitochondria, by the way, in the cell? Mitochondria, remember, divides independently by a cell cycle by simple fission. Okay? It has also your DNA and 70S ribosome. That is why, class, the origin of your mitochondria is linked to the origin of prokaryotic organism. So, mitochondria cannot be made from a scratch because they need both both mitochondrial and nuclear gene products. These organelles replicate by dividing into two using a process called simple fission. Similar with your bacteria, mitochondrial division is stimulated by energy demand, so the cell with an increased need of energy contains greater numbers of these organelles. So meaning, the more energy the cell uses, the more po siya merong maraming mitochondria. But if the cell requires lower energy, therefore, it has only few mitochondrial organelles. Another cytoplasmic structure is your plastids. Your plastids are responsible for photosynthesis. Your plastids, by the way, are only found in plants. Okay? They are storage of products like starch, and for synthesis of many classes of molecules such as fatty acids, terpenes, which are essentially needed for building blocks or for functional production for plants. So, your chloroplasts are one type of plastid, contains green photosynthetic pigment called chlorophyll. So your chloroplasts are found in plants again and algae. So your plastids class is the same with your mitochondria. It has also its own DNA and ribosomes. So which these DNA and ribosomes are covalently close circular form that codes for some of the constituent proteins and transfer RNA. So later on your readings class, I want you to find out what's the difference between plastids and ribosomes, and plastids and mitochondria, since they contain both DNA and ribosome structures. So another cytoplasmic structure is your lysosomes and your peroxisomes. So these two structures are different. Okay, your lysosomes contains degradative enzyme called lysozyme that breaks down foreign materials taken into the cell by phagocytosis. So these enzymes aids in breaking down and worn out parts of the cell and may destroy the entire cell process called autolysis if the cell is damaged or deteriorates. So your lysosomes are found in all eukaryotic cells. Your peroxisomes naman contains an enzyme called catalase. So your peroxisomes are membrane-bound vesicles in which hydrogen peroxide is both generated and broken down. So it contains oxidative enzyme, your catalase, which generates ATP production, which helps in generating ATP production, and your catalase also speeds up or catalyzes the breakdown of hydrogen into water. So your peroxisomes is commonly found in mammalian liver cells. So again, the main difference between lysosomes and peroxisomes, lysosomes class are degradative while peroxisomes are oxidative. Okay? So when we say degradative, it does not produce energy or does not help energy production, while your peroxisomes, since it is oxidative, so it may help in generating energy production or during ATP synthesis. Last cytoplasmic structure is your cytoskeleton. So your cytoskeleton is a three-dimensional structure that fills the cytoplasm or systems of fibers. Okay? So, you have three types of fibers for your cytoskeleton. You have your microfilaments. Microfilaments with a diameter of 3 to 6 nanometer. So, primarily, your microfilaments class is for cell defining and maintaining cell shape. It also functions during um, motility like gliding and contraction, muscle contraction, and cytokines. Okay? So you have your intermediate filaments. Intermediate filaments class um, measures 10 nanometer in diameter and commonly function for tensile strength of the cell. And lastly, microtubules are cells responsible for cell structure, cell motility, and cell division. So all three types of this cytoskeleton class in general serve to strengthen, support, and stiffen the cell, and it gives the cell shape. In addition, your microtubules and your microfilaments class are essential for various activities like cell division, contractions, or motility. The cell division mentioned by the way, in which microtubules assist microfilaments in maintaining cell structure to form spindle fibers for separating chromosomes during mitosis. So another structure is your plasma membrane. So your plasma membrane is also known as your cell, cytoplasmic, or your cellular membrane. So your plasma membrane acts as a skin around the cell. So the cell membrane consists of lipid bilayers that is semi-permeable. The cell membrane also regulates transports of protein materials entering and exiting the cell. So structurally, your cell membrane or your plasma membrane is a mosaic composed of large molecules of proteins and phospholipids. So these phospholipids are your certain types of fats. The cell membrane regulates the passage of nutrients, waste products, and secretions into and out the cell. Because the cell membrane has a property of selective permeability, only certain substances class may enter and leave the cell. The cell membrane is also similar in the structure and function to all of the membranes that are found in eukaryotic organisms. So cell wall. Your cell wall, some eukaryotic cells contain cell wall. These are external structures that provide rigidity, shape, and protection. Eukaryotic cell wall, which are much simpler in structure than your prokaryotic cell wall. It may contain cellulose, pectin, lignin, chitin, and some mineral salts which are usually found in algae. So the main function of your cell wall class is to provide protection to the internal structure of the cell. Since the plasma membrane is considered to be a fragile layer that cannot provide similar protections against different environment conditions, additionally, cell walls are responsible for providing distinct shapes of the cell. When the internal pressure inside the cell increases due to the entrance of water, the cell wall prevents expansion and rupture of the cell. So the cell wall mainly controls the passage class of the molecules across the cell, allowing only small metabolic molecules to pass. In multicellular organisms, the cell walls may help in adhesion of cells, giving them distinct shapes. So the cell walls of algae and plants class are made of primarily cellulose. Okay? Cellulose is also found in some of the structures of prokaryotic organisms. The cell wall of fungi is made up of polysaccharides chitin, which is not found in the cell wall of any other microorganisms. Chitin is similar. It has also similar structure to the cellulose found in the exoskeletons of your crabs. Okay? For motility organelles. So for motility, you have two basic structures. You have the cilia and your flagella. Cilia class and flagella is different, but these two may help for locomotion. So your cilia, on the left side, these are short hair-like appendages found only in eukaryotic organisms like your paramecium or your Balantidium coli. If you still remember your parasitology, the only ciliated protozoan pathogenic to human. So primarily, the movement of your cilia is sweeping or pendular movement. So your cilia is not only for locomotion, but it is also used for feeding and circulation. Your flagella, these are long whip-like structures. These are found in both prokaryotic and eukaryotic organism. This may be found throughout the cell or either at the one end portion of the cell. So we'll discuss more of this when we go to prokaryotic organism. So the main movement of your flagella class is undulatory movement. The same movement with some snakes, di ba? Kanang pa-side sweep siya. So the main function of your flagella is for locomotion. Remember, class, that both flagella and cilia of eukaryotic cells have the same basic structure and biochemical composition. Both consist of series of microtubules, hollow protein cylinders, composed of protein called tubulin, surrounded by a membrane. The arrangement of the microtubules class is called 9+2 system. Again, the arrangement is called 9+2 system. Why 9+2 system? Because it consists of 9 peripheral pairs of microtubules surrounding two single central microtubules. So, 9 peripheral pairs of microtubules surrounding two single central microtubules. So, that's and the eukaryotic side. Now, let's go to prokaryotic cell. So, for prokaryotic cell, most likely, class, they have the same structures, but limited only. The same structure, the same functions, but limited lang siya. Okay. So, prokaryotic organisms, you have the most common, your bacteria and your archaea. Bacteria class are unicellular organisms that do not have true nucleus, a nuclear membrane, and a membrane-bound organelles. That is why they are called prokaryotes. Pro means before, karyons means nucleus, nuts, or kernel. So, the image shown are structural features of composite idealized bacterial cell and electron micrograph of an actual bacteria, which is your E. coli. If you've noticed, the structures highlighted in the red are found in all bacterial species. So, in all bacterial species, you have cytoplasm, plasma membrane, ribosomes, and nucleoid containing DNA. So, the rest, class, may or may not be found in some bacteria. So, your prokaryotic cell structures are subdivided into three. You have there the external structure, the cell envelope, and the internal structure. So, let's start with the cell's internal structure. You have the cytoplasm, the nucleoid, ribosome, storage granules, and plasmid. So, your cytoplasm class or protoplasm of a bacterial cell is a gel matrix composed of water, enzyme, nutrients, waste, gases, inorganic ions, and many low molecular weight compounds. Okay? Your cytoplasm also carries the function for cell growth, metabolism, and replication. So, since it carries this function class, your cell cytoplasm contains your ribosomes, chromosomes, and your plasmids. So, another intracellular structure, you have your nucleoid and bacterial chromosomes. So, unlike eukaryotic cells, bacteria do not have membrane-enclosed nucleus. That is why they are called prokaryotic organism. So the nucleoid, or meaning nucleus-like, is an irregular shaped region within the cell of a prokaryotic that contains all or most of the genetic materials. In contrast to the cell nucleus of your eukaryotic cell, it is not again surrounded by a nuclear membrane. So the genome of the prokaryotic organism generally is a circular, double-stranded piece of DNA which multiple copies rather, may exist at any time. So not all class are circular double-stranded DNA. There are also bacteria class that contains linear type of DNA, like your Borrelia, Burgdorferi, and your Streptomyces species. So these organisms contains linear type of chromosomes. The nucleoid, additionally, the nucleoid can be clearly visualized on electron micrograph at a higher magnification. Sometimes even stands out of what is thought to be DNA are visible. So the nucleoid can be seen under a light microscope using fulgen staining. So fulgen. So we'll just discuss more of it as we go along. Some textbooks or studies class specify prokaryotic chromosomes as genophore. The term chromosomes sometimes is misleading because the genophore lacks chromatin. The genophore is compacted through mechanism known as supercoiling, but chromosome is additionally compacted through the use of chromatin. The genophore is circular in most prokaryotes and linear in some of the few. So overall class, prokaryotic DNA interacts with nucleoid-associated proteins that assist the organization and packaging of chromosomes. In bacteria, these proteins function similar to histones in your eukaryotic organisms, which organize DNA in eukaryotic cells. So the image shows the prokaryotic cells on the right and eukaryotic cells on the left. Comparison and electron microscopy of your E. coli. Another intracellular structure is your plasmid. So prokaryotic cells may contain extrachromosomal DNA or DNA that is not part of the chromosomes. This extrachromosomal DNA is found in plasmids, which are very small, circular, and double-stranded DNA molecules. They can replicate independently of chromosomal DNA and transmit genes encoding for drug resistance, bactericin, toxins, and more from one bacterium to another via conjugation and transduction. So you have 5 types of bacterial plasmids. You have your fertility plasmids or your F plasmid, which is capable of conjugation. When we say conjugation, this is a transfer of genetic information between 2 cells. They are not involved in cellular reproduction. Again, conjugation is different from cellular reproduction. So the main responsible for responsible for conjugation is your PILI or FIMBRIAE, which will be discussed later. Resistance plasmid or your R plasmid are also known as R factor, contains antibiotic or drug resistance gene. Okay, that is why it's known as resistance. Your virulence plasmids, the main function of virulence plasmids, it turns the host of the plasmid or the bacterium into pathogenic. So if ever the organism contains this type of plasmid, instead na hindi siya pathogenic, it will not cause disease. Once it has the virulence plasmid, so it may now cause the disease. Okay? Degradative plasmids, the main function, it enables digestion of unusual substances like toluene or salicylic acid. So it degrades structures. Your col plasmid contains genes that code for proteins that can kill other bacteria. So common example for plasmid is your bacteriocin, which is commonly seen in E. coli. So the image shows your plasmids. There, the circular double-stranded structures below, and above class, that is how plasmids are passed to bacteria. So they can be integrated into chromosomal DNA during conjugation, or just simply replicate and pass the genes on the daughter cells during binary fission. So I guess you'll have a separate lectures for cellular genetics on the succeeding lectures. So ribosomes. All cellular life synthesize proteins and organisms in all three domains of life processes. And ribosomes is the structure responsible for that protein synthesis. Ribosomes is also known as polyribosomes or polyosomes. Polyosomes are a group of ribosomes bound to mRNA that collectively synthesize proteins. So your ribosomes translate the genetic code from the molecular language of nucleic acid to that of amino acid, which is your building blocks proteins. Proteins are the molecules that perform all the functions of cell and living organisms. Ribosomes themselves are constructed from proteins alongside with your ribosomal RNA. So, the ribosomes are composed of hundreds, thousands, and found in the cytoplasm that gives it granular appearance when viewed under microscopes. So, your prokaryotic, again, ribosomes are called 70S ribosomes because they have a size of 70S composed of two subunits, small subunits of 30S and large subunits of 50S. So again, prokaryotic 70S, eukaryotic 80S. So the S there stands for the Spidberg unit, which is a measure of sedimentation in an ultracentrifuge, which is based on size, shape, and surface qualities of structures. So many antibiotics class binds to either 30S subunits or 50S subunits of your bacterial ribosomes. So inhibiting the function of bacterial ribosomes, but not the eukaryotics. So for example, you have your streptomycin. Your streptomycin binds the 70S ribosomes, particularly the 30S subunits. So it stops the protein synthesis, and therefore there will be no protein production, but it will not stop the protein synthesis of the eukaryotic because it is only limited to 70S. Si 70S lang yung kaya niyang i-bind, but yung 80S hindi. So, your tetracycline binds to your 30S subunit still. By the way, your streptomycin antibiotic is commonly treatment for TB. Your tetracycline, usually a broad-spectrum antibiotic, so it can kill gram-positive and gram-negative. Your chloramphenicol also binds to 30S subunits. Linezolid binds to the initial step process of the protein synthesis. So your linezolid is commonly used for gram-positive organisms. So you'll get to know more about antibiotics and their mechanism class on a separate lecture. Inclusion bodies. As single-celled organisms living in an unstable environment, some prokaryotic cells class have the ability to store excess nutrients within cytoplasmic structures. And these structures, cytoplasmic structures, are called inclusion bodies. Inclusion bodies do not possess any metabolic activity. So, some inclusion bodies are called insoluble granules, others are called vesicles. So, the main purpose is to store nutrients or food reserves for cellular functioning. Your inclusions are non-living components of the cell. The most common type of inclusions is your poly-B hydroxybutyric acid or your PHB. Your PHB is a um, lipid-like compound formed when the source of nitrogen class or sulfur or phosphorus is limited and there is excess in carbon in the medium. So, other inclusion bodies, you have your glycogen, sulfur, phosphates, polyphosphates rather, or called as your volatile or metachromatic granules. So your vesicles are rigid gas-filled vacuoles which is commonly found in aquatic photosynthetic bacteria. And your inclusion bodies, by the way, also serve as important identification characters for some bacterial pathogens like your Corynebacterium diphtheriae. So your Corynebacterium diphtheriae class contains an inclusion bodies known as Babes urn granules or Babes urn bodies. So these Babes urn granules or bodies are made up of metachromatic granules or volatile granules. So these granules can be degraded and used as source of phosphates for nucleic acid and phospholipid synthesis to support its growth. So they are termed volatile or metachromatic because they usually stain red with a blue or green dye. So again, take note, the name of the Corynebacterium diphtheriae inclusion bodies is Babes urn granules. Now let's go to cell envelope. The cell envelope class is a complex structure that forms two wrappers, the cell wall and your cell membrane which surround your cytoplasm. Your cell wall is relatively porous to the movement of the substances, whereas your cell membrane regulates the transport of nutrients and metabolic products. Additionally, an outer membrane that is found only in gram-negative cells. So let's start with plasma membrane. So your plasma membrane class similar functions with your eukaryotic plasma membrane. So your plasma membrane is also known as your cell membrane or cytoplasmic membrane, which is a biological membrane that separates the interior of the cell from its outside environment. So the primarily function is to protect cell from its surrounding. So, the plasma membrane structure of the most bacterial organism is a bilayer composed of mainly phospholipids formed with ester linkages of proteins. These phospholipids and proteins have the ability to move laterally within the plane of membranes as well as between two phospholipid layers. So your plasma membrane also is the site of metabolic reactions per prokaryotic organisms like respiration, fermentation, and photosynthesis. So the substances, by the way, class on your plasma membranes move either passive or active or group translocation. So passive, it occurs without input cellular energy. Active, the transport requires expended energy, while group translocation, the solutes move without change in structures. So for this mechanism, passive, active, and group translocation class, I want you to review the transport mechanism of substances, say, in your biochemistry lectures. So your plasma membrane also involves in excretion of hydrolytic enzymes and pathogenicity proteins like your protein secretion systems and chemotactic agents. In short, class, your plasma membrane, think of it as a castle. If the plasma membrane is the wall that provides structure for the building inside, which regulates people live and enter the castle and conveys message to and from their neighboring castle. So, just a hole in the cell wall can be disaster to the castle. So a rupture of the plasma membrane causes the cell to lysis and die. So that is why most of the antibiotics, there are some antibiotics class that targets plasma membrane. Right? Your cell wall also is the target of some antibiotics like your penicillin and some cephalosporins. So penicillin kills bacteria through binding the beta-lactam ring to the DD transpeptidase, inhibiting the cross-linking activity and preventing new cell formation. So without a cell wall, a bacterial cell is vulnerable outside conditions like water or molecular pressures which causes cell to quickly die. Since human cells class do not contain cell wall, penicillin treatment or any cell wall inhibitor treatments result in bacterial cell death without affecting human cell. Propeptidoglycan inwards of the cell are similar. Going further out, the bacterial world divides into two major classes. So, you have the gram-positive and gram-negative organism. So, amount and location of the peptidoglycan in the cell wall determine whether a bacterium is gram-positive or gram-negative. And this classification is because of a differential staining called gram staining. So what is differential staining, by the way? Differential staining, just to give you a definition, is a staining process which uses more than one chemical stain. So using multiple stain can better differentiate between different organism or structures or cellular components of a single organism. So your gram staining is the most common differential stain. This is introduced by a Danish bacteriologist, Hans Christian Gram, in 1884. So, your gram staining differentiate bacteria into gram-positive and gram-negative organisms. So, when bacteria is stained with primary stain, your crystal violet, which is your crystal violet, and fixed by the mordant of the some bacteria are able to retain primary stain and some are decolorized by alcohol. So the cell wall of gram-positive bacteria, since it is thicker, it contains protein sugar complexes called peptidoglycan and the lipid content is low. Decolorizing the cell causes this thick cell wall to dehydrate and shrink, which closes the pores in the cell wall and prevents the stain from exiting. While for gram-negative organism, since it has lipid component and thinner peptidoglycan layer, when the CVI complex or the CVI iodine complex wash off due to the decolorization process, it dissolves the lipid components of the cell wall, which allows the crystal violet iodine complex to leach out the cell. So therefore, class, since the major component of your gram-negative is lipid, so when you add decolorizer, like your alcohol or your acetone, the CVI complex formation after the additional of your crystal violet and your mordant, it will leach out the cell, giving your organism to be unstained. So when you put a counter stain, obviously it will accept the counter stain color. While your gram-positive, since there will be no pores already because it will shrink, it will dehydrate when you decolorize. So therefore, the counter stain will not be received or will not be poured out into the cell. So your gram-positive organism will stain dark blue or violet because of the primary stain, while your gram-negative organism will be stained pink because or red in some textbooks because of the secondary stain, which is your safranin. So we'll not discuss more of this since you're not medical technologists and you'll not be dealing this in the lab. So this is a picture of a gram-negative bacteria and a gram-positive bacteria. Okay, so as mentioned, your gram-negative organism contain an extra layer prior to the cell wall. So your outer membrane class is the second lipid bilayer found in between the cell wall and the plasma membrane. Your outer membrane is located external to your peptidoglycan layer. So here in the picture, this is your peptidoglycan layer on the blue part, and above that is your outer membrane structure. The outer membrane is attached to the peptidoglycan layer by murine lipoprotein or bron lipoprotein. And at one end, the lipoprotein is covalently bound to the peptidoglycan layer, while the other end is embedded into the outer membrane via its polar head. So you have three structures that composes your outer membrane. You have your O antigen or your O polysaccharide, your core polysaccharide, and your lipid A. So your O antigen or your O polysaccharide represents the outermost part structure. Okay, while your core polysaccharide and lipid A anchors the lipopolysaccharides into the outer membrane. So your lipopolysaccharide is known to serve many different functions, such as it contributes to the net negative charge of the cell, helping to stabilize the outer membrane and providing protection from a certain chemical substances by blocking access to other parts of the cell wall. In addition, your lipopolysaccharides are role in the host response to the pathogenic gram-negative bacteria. The O antigen class triggers an immune response in an infected host, causing generation of antibiotics specific to that part of lipopolysaccharide. So if you have read E. coli O157, the O there is a side chain called antigen that can be detected using serological or immunological tests to identify specific strain of E. coli. This E. coli O157 class is a type of E. coli that can cause hemolytic uremic syndrome. Okay? It's a deadly strain of bacteria that causes bloody diarrhea, kidney failure, as I said, while your lipid A class acts as a toxin. So your lipid A is an endotoxin. It's a type of endotoxin, specifically causing general symptoms of illness such as fever and diarrhea. That is why, class, most of the gram-negative organisms are pathogenic yun na siya because of this lipid A component or your endotoxin. A large amount of lipid A release into the bloodstream that can trigger to endotoxic shock, which can cause a body-wide inflammatory response, which can be life-threatening if not treated. Aside from gram-negative and gram-positive cell wall, you also have acid-fast cell wall. Your acid-fast cell wall is primarily found in Mycobacterium species. So, it contains large amounts of waxes. So, your waxes are complex branch of hydrocarbons, primarily known as mycolic acid. So, these are common in tubercle bacillus or your Mycobacterium tuberculosis. So why it is named as acid-fast? Because the principal class, if a dye is introduced into this cell by brief heating or treatment with a detergent, it cannot be removed by dilute acid. But when we do that in a simple organisms like E. coli and Staphylococcus, then these organisms will be stained, you say, for your primary stain. So the cell wall is composed, class, for acid-fast cell wall, by the way, the cell wall is composed of peptidoglycan and external asymmetric lipid bilayer. The inner leaflet contains mycolic acid linked to an arabinoglycan. So if you go here to the structure, and outer leaflet contains other extractable lipids. This is highly ordered lipid bilayer in which proteins are embedded, forming a water-filled pores through which nutrients and certain drugs can pass slowly. Some compounds can also penetrate the lipid domains of the cell wall a bit slowly. These hydrophobic structures render these bacteria resistant to many harsh chemicals including detergents and strong acids. So, primarily, your acid-fast organisms class are resistant to some chemicals. That's why it will require intense disinfection or sterilization. You also have organisms class that do not contain cell wall, and these are cell wall deficient bacteria. So, common example, you have Mycoplasma. Your Mycoplasma genomic analysis, it belongs to a gram-positive organism. So, since these organisms do not contain cell wall, so it lacks target cell wall inhibiting agents. Okay? So, treatment. The treatment should not be done using cell wall inhibitors, antimicrobial, like your penicillin, because these are resistant to penicillin. So, common example of your Mycoplasma includes your Mycoplasma pneumoniae, which is an agent of atypical pneumonia. So now let's go to external structures, your flagella, pili, and your glycocalyx. So to begin with, your flagella. Flagella are hair-like structures that provides means of locomotion for those bacteria that have them. Not all naman organism or bacteria class in general contains flagella because there are some bacteria do not have flagella. Therefore, they are atrichous or absent of flagella. So, the flagella beats in propeller-like motion to help the bacterium move towards nutrients, away from toxic chemicals, or towards the light, like your cyanobacteria. Okay? That is why the movement of your organism for this one is angular movement. Each flagellum is attached to a cell membrane with the help of proteins other than flagellin. Okay? So the basal body of the flagellum is embedded in the plasma membrane. A hook region connects the basal body to the filament. The gram-positive and gram-negative bacteria have different basal body configuration due to the differences of the cell wall. So, the basal body consists central rod or shaft surrounded by set of rings. For gram-positive, take note, it contains 2 basal rings, while your gram-negative contains 4 basal rings in the flagellar structures. So, primarily, again, flagella is for locomotion lang yun na siya class. So, bacterial species exhibit different arrangement and numbers of flagella. So, a bacterium with a singular flagellum, typically located at one end of the cell or the polar region, is said to be monotrichous. An example of monotrichous organism, you have your Vibrio cholerae, which is a gram-negative organism that causes cholera. Cells with amphitrichous flagella have flagellum or tuft of flagella at each end. So, common example, you have Spirillum minor, which causes spirillary or Asian rat bite fever or Sodoko. Cells with lupotrichous flagella have tuft at one end of the cell. So, common example for lupotrichous flagella is your Pseudomonas aeruginosa, an opportunistic pathogen known to cause many infections including swimmer's ear and born-borne infections. Flagella that covers the entire surface of the bacterial cells are called peritrichous flagella. So the most common example here is your E. coli or your Escherichia coli. Again, since this is microbiology class, you need to be specific. You need to write down the name Escherichia, Escherichia coli, because again, you have two E. coli in microbiology world. You have your Entamoeba coli and your Escherichia coli. Another cellular appendages is your Fimbriae and PILI. Your Fimbriae and PILI are structurally similar, and because differentiation between the two is problematic, these terms are often used interchangeably. Without PILI, many disease-causing bacteria lose their ability to infect because they are unable to attach the host. So your Fimbriae class is a short bristle-like proteins projecting from the cell surface by hundreds. So the primary functions of Fimbriae is for attachment to the surface and to the other cell, while your PILI is longer, less numerous protein appendages that aid in attachments of surfaces as well for conjugation. So you have two types of PILI, your F. pili or your sex PILI is important in the transfer of DNA bacterial cell. As mentioned, you have your F. plasmid and your type 4 PILI, which generates movement. The movement is called twitching motility, where pilus attaches to a solid surfaces and then constructs by pulling the bacterium forward in a jerky movement. So picture is an example of how PILI, how organism transfer its genetic materials to another organism. So your Fimbriae, these are the short structure, while your PILI are the longer structure. So lastly, you have your glycocalyx as external structure. Your glycocalyx are sticky coating produced by many bacteria covering the surface of the cell. Functions is for protection, attachments to the surface, and formation of biofilms. So you have two forms of glycocalyx. You have your capsule, which is a polysaccharide layer that completely envelopes the cell. It is a well-organized and tightly packed, which explains its resistance to staining under the microscope. The capsule offers protections from a variety of different threats to the cell, such as desiccation, hydrophobic toxic materials, and bacterial viruses. The capsule also enhances ability of bacterial pathogens to cause disease and can provide protection from phagocytosis. So phagocytosis is engulfment of white blood cell. And lastly, your capsule helps in attachments to the surfaces. While your slime layer class is similar to capsule that is typically composed of polysaccharide and it completely surrounds the cell. However, class, your slime layer is disorganized through structures. It also offers protections from various threats such as desiccations and antibiotics, and also allows adherence to the cell surfaces. So here is a picture of your capsule glycocalyx, which is a structured and well organized, and your glycocalyx slime layer, which is disorganized. So an additional structure is your endospores. Your endospores are dormant, tough, non-reproductive structures that undergo germinations or vegetative cell. You have two common genera that forms endospores. You have your Bacillus and Clostridium. And take note, class, this Bacillus and Clostridium class are both gram-positive organisms. So these organisms class undergo a cycle of differentiation in response to environmental conditions. So the process of sporulation is triggered by a near depletion of any several nutrients like your carbon, nitrogen, and phosphorus. If there is depleted nutrients class, it forms spores. So each cell forms single internal structure that is liberated when the mother undergoes autolysis. So endospores are resistant to UV and gamma radiation, desiccation, lysozyme, enzyme, temperature, starvation, and chemical disinfectant. However, endospores are killed using autoclaving. So that is why for spore-forming bacteria or spore-forming organism, the only way to kill that is using autoclave. So the spore is a resting cell also. So when the organism receive appropriate nutrients class, it will return to favorable nutritional conditions and it will be activated. So the spore germinates to produce a vegetative single cell. So sporulation, meaning indoor spore formation, and when the spores already gain those nutrients that is depleted, it will undergo naman germination. So it will return to its active state or vegetative state. So the process here is a sporulation process. So this is an example of how Bacillus subtilis undergoes sporulation. So in response to starvation, so as I said, if there is depleted nutrients like carbon, nitrogen, and phosphorus, so the Bacillus subtilis differentiates into dormant stress-resistant cell type known as spores. So the cell initiates spore formation with an asymmetric cell divisions that generates a large cell and a small cell or your spore or your prospective spore. Initially, these two cells, the side by side, but later in development, the mother cell engulfs the spore in a phagocytic-like process to produce a single cell within a cell. The mother then nurtures the spore as it prepares for dormancy. Once the spore is fully mature, the mother cell lyses, and we use this elegant the mother cell lyses, and it will now form into another endospore. So the picture here is an example of how do we view endospores in the lab. So the green ones are your endospores within a pink bacilli. So many spores have already been released from vegetative cell. So those organisms class without green structures in between are vegetative cell. But those organisms with green structures in between them are endospore-forming cell. Okay? So spores can be located either central, terminal, or subterminal. And these locations can also be used in identifying microorganisms. So to wrap up, this table here summarizes the salient points about bacterial cell structure, its function, and some additional information related to each structure. So you can read this one as summary of the lectures I have discussed. So another important topic to note is the cellular division. So again, your microorganism, particularly bacteria, undergo cellular division called binary fission, which is an asexual reproduction. So cell growth and reproduction by cell division are tightly linked in unicellular organism. So in growing culture like E. coli class, cell elongates and then form into partition. So if you look the image on the left or on the right side, so think of it as E. coli, the cell elongates and then form into partition and then eventually separates the cell into two daughter cells. The partition is referred to as a septum, okay, or a cross wall. And its results is inward growth of cytoplasmic membrane and the cell wall from opposing direction until the two daughter cells are pinched off. The chromosomes, which have doubled in numbers preceding the divisions, are distributed equally to the two daughter cells. Under normal condition, bacteria can grow and divide extremely rapidly, and bacterial population can double as quickly as every 9 to 8 minutes. So, for this process class to occur, cell division, a cell must grow over its entire surface until the time of cell division. Okay, for the cell to divide in half, the peptidoglycan structure must be different in the hemispherical cap than in the straight portion of the cell wall. And the different wall cross-linking enzyme must be active at the septum than elsewhere. So these are the two processes that should be taken note when during binary fission. So in general class, microorganism again replicates through binary fissions, particularly your bacteria. So your bacteria, if the cell remains temporarily attached after division, certain characteristics groupings result depending on the plane of division and the number of division through which the cell will remain attached. The following may occur into coccal forms like chain, which we call as streptococcus, pairs, which we call as diplococcus, cubical bundles, which we call as sarcina, or flat plates like your rods. Your rods may also form in pairs and in chain. After fission of some bacteria, there are characteristic post-division movements. So for example, class, a whipping motion can bring up the cells into parallel positions, repeated divisions, and whipping results like your palisades, like this one, Chinese little patterns, which is a characteristic of a diphtheria bacilli. So the picture here is how the coccus and the bacillus divide. So the plain divisions, if the cocci divides into two, you have your diplococci. If the cocci divides into chain, you have your diplococci. If it divides into four, you have the tetrads, into eight, sarcina, and into irregular patterns, you have your staphylococci. The same with your bacillus. If the bacillus divides into two, you have your diplobacilli. Into chain, you have your streptobacilli. So here are listed references. So you can go over the references if you want to have supplementary readings. So most common class, I get the topics from Jawitz, since Jawitz is the main book that we will be using for medical micro or clinical micro. So this, you can read this one in chapter 2 of Jawitz. So questions, clarifications. So any questions or clarifications, feel free to drop me an email at this email address, or you can chat me on my Facebook account. So you can search my name sa Facebook for questions and clarifications. So I hope you learned something, and we'll go to the next lesson. Thank you.